We have is Meera Krishnamoorthy. Apologies, the name is not correct. She is a third-year PhD student in computer science at the University of Michigan. She received her bachelor's degrees from Caltech with major in electrical engineering and minor in computer science. She now works with Professor Gina Wins on developing novel machine learning techniques to classify healthcare data. Do please give a warm welcome to Meera.
Hi, everyone. My name is Meera, and today I'm going to be presenting on my work entitled AMAISE, which stands for a Machine Learning Approach to Index-Free Sequence Enrichment. And this work was published in Nature Communications Biology in June of this year. And it was done with Piyush Ranjan, John Erb Downward, Robert Dixon, and my thesis advisor, Jenna Weens. And I just want to preface this by saying that I adapted this talk from some slides that I made for primarily a computational audience. So it might be a little too basic for people that have more of a biological background, but hopefully you'll still enjoy. Okay. So the overarching goal of this work was to improve infectious disease diagnosis, and specifically to help to move towards a pathogen identification tool that is accurate, fast, and cheap and efficient. So accurate to ensure correct treatment, fast to ensure treatment can be given in a timely manner, and cheap and efficient to ensure the accessibility of the method and the ability of it to be used at the bedside. So traditionally, infectious diseases are diagnosed using culture-based methods. And so in doing so, clinicians will typically take a sample from a patient, that could be stool, saliva, or blood, mix the sample with culture media and wait for growth, and then use biological tests to perform the pathogen identification and classification. And so two drawbacks of these methods can be that one, these methods can be inaccurate because not all pathogens can grow mixed with the culture media, so they can't all be detected using this method, and detection is necessary for treatment. These methods can also be slow because it can take several days to detect growth, and that can negatively affect a patient's ability to recover. So more recently, people have been moving towards using metagenomics for disease diagnosis. And so metagenomics is the study of the metagenome, or the collective DNA from a clinical sample. So to use it to perform disease diagnosis, clinicians can take that same sample from a patient, but then input it into a DNA sequencer. And the output of that, the metagenomic sequencing output, is all of the DNA from the sample. And then they can use metagenomic classification methods to classify the pathogens in that sequencing output. And so metagenomic classification involves mapping DNA to a classification label. And existing metagenomic classification methods do this using the reference database that you see on top there. And so the problem with metagenomics for disease diagnosis are that there can be copious host-derived sequences in samples. So this is what a lot of samples that you can get from a patient look like. And so in blue, we have the microbial sequences, which are informative for pathogen detection because they could contain that pathogen-derived sequences. Whereas in red, which occupies a lot of the sample, are the host sequences, which are not informative for pathogen detection. And in this case, host is the patient from which the sample was collected. So copious host-derived sequences can cause downstream classification methods to be inaccurate because these methods aren't especially sensitive to host sequences, especially when these sequences are noisy. It causes them to be slow because they have to classify a lot of uninformative sequences in the hope of getting to a pathogen-derived sequence, as well as costly because a lot of host genomes can be large, so you have to store all of these to perform that classification. So to alleviate the aforementioned problem, prior investigators have employed host depletion steps. And so host depletion is a binary classification task of determining whether a DNA sequence is from a host or not. So in order to use host depletion, instead of this kind of typical pipeline where you have your DNA, you input it into a metagenomic classification method, which will use a database with both host and microbial data to perform the pathogen classification. You can employ a host depletion method before that metagenomic classification method that'll remove the host sequences, inputting the microbial sequences only into the metagenomic classification method. And as you can see here, the database that that method will need is a little bit more lightweight because you don't have to store those host genomes anymore. So the benefit of using host depletion are that it allows downstream methods to be more accurate because it gives them a slightly easier classification task, faster because they have to classify less sequences, and cheaper because they don't need to store those large host genomes. Okay, so previous work in computational host depletion can kind of be broken up into two different groups. One is called pairwise alignment. An example of this type of method is Minimap2, where a query sequence is aligned to a host reference genome. And in this case, our host is human. As you can see here, the query sequence perfectly aligns, so it would output that that query sequence is from the host. The other type of host depletion method are lookup tables like Kraken2 and Centrifuge, where you have your query sequence that's broken up into k-mers or subsequences of length k. In this case, k is 3. And then each of those subsequences is mapped directly to a subsequence in a database, and that label is assigned to the query sequence. And as we can see in this case, all of these subsequences are mapped to the human, and so we output a human classification label. So the issue with these type of methods are that they can be inaccurate on sequences from error-prone technologies because they're building their reference databases typically using reference genomes, which are very curated and have very few errors in them, unlike sequencing output that can have sequencing errors in them. And also these methods can be costly because they require the storage and memory to store reference databases. So why do we think machine learning could help? So we thought that machine learning would allow for more accuracy as well as cost effectiveness. For accuracy, we hypothesized that an ML-based approach would be more robust to sequences from error-prone technologies because we can build a training set with reference genomes and sequences from error-prone technologies. We also hypothesized that an ML-based approach would use less storage and RAM than existing host depletion methods because we can learn patterns from raw DNA sequences without the reliance on an external database. So our contributions are that we developed a novel machine learning-based host depletion method, AMAISE, that can perform host depletion more accurately and cheaper than existing approaches. And we rigorously evaluated our approach by first providing a comprehensive comparison of our approach and existing alignment and classification methods in their ability to perform host depletion. And we also evaluated our approach as a preprocessing tool applied before downstream microbial taxonomic classification to see what the gains are using AMAISE in the entire pipeline. So we used 5 test sets for our evaluation, and each test set varied in the percentage of reads of the sample that pertain to host. We go from 1% to 99%. We used human in our host fraction and bacteria and fungi in the microbial fraction, and each of our test sets contained nanopore reads. And we focused our evaluation on nanopore reads because nanopore sequencing currently produces the longest reads and is the only platform that produces real-time sequencing data, both of which are useful for clinical diagnostics. So we had some different evaluation metrics that we were looking at to look for accuracy, speed, and cost and efficiency. So accuracy, again, is important for ensuring the correct treatment. For our host depletion comparison, we looked at general classification accuracy as well as sensitivity and specificity, where sensitivity is the percentage of host DNA that was correctly identified. Specificity is the percentage of microbial DNA correctly identified. And then for our preprocessing / metagenomic classification comparison, and this is a multi-class task, we looked at overall accuracy on the host data and microbial data. And then for speed, we looked at wall clock time, and cost and efficiency, we looked at storage, VRAM, and RAM usage. So for our first comparison, we compared AMAISE's ability to perform host depletion to other computational host depletion methods, where our hypothesis was that AMAISE would be more accurate and cheaper than k-mer and alignment-based approaches at performing host depletion. So just again, for the schematic workflow of how we were using host depletion, for each method, we inputted the metagenomic sequencing output, and we looked at how well those methods were able to separate the host sequences from the microbial sequences. And the baseline methods we were comparing AMAISE to are Kraken2, Centrifuge, and Minimap2. And all of these methods just had host DNA in the reference databases. So first, I'm going to be presenting our results on accuracy. We have host fraction on the x-axis, and then accuracy, sensitivity, and specificity on our y-axis. AMAISE's performance is in purple. The other methods are in different colors. And for all of these metrics, higher is better. And you can see across host fractions, AMAISE consistently achieved a higher accuracy and sensitivity, as well as comparable specificity compared to all of our baselines. Now looking at cost, in this case, lower is better. AMAISE is still in purple. We see that AMAISE required less storage and as well as remained competitive with respect to peak memory usage compared to baselines and performs a lot better than Minimap in green. And then for classification time, we see for speed where lower is better again. AMAISE remains competitive with respect to speed compared to baselines and especially is faster than centrifuge, whose speed gets much worse as the host fraction percentage increases. Okay, so now moving on to our metagenomic classification comparison, where we compared the metagenomic classification pipelines with and without AMAISE. And our hypothesis was that AMAISE would improve the accuracy and efficiency of metagenomic classification methods. So just as a reminder of the comparison that we're making here, we're comparing the pipeline highlighted in orange to the pipeline in purple, where in orange, we're just inputting DNA into a metagenomic classification method. In purple, we use AMAISE before the metagenomic classification method. And here we focus our comparison on centrifuge. So it's AMAISE plus centrifuge and centrifuge alone. And we focused on our comparison with centrifuge because we found that centrifuge actually outperformed Kraken on every metric except for classification time. So first, I'm going to be presenting my results on accuracy. So in this case, we have in pink centrifuge alone, in purple AMAISE plus centrifuge. For accuracy, again, higher is better. We see that the pipeline that included AMAISE consistently achieved a higher host accuracy. In terms of peak memory usage, where lower is better, AMAISE reduced the peak memory usage requirements for centrifuge. And then speed, again, where lower is better. The pipeline with AMAISE's classification time decreased as the percentage of host data in the test set increased. And then the pipeline without AMAISE's classification time increased. And for most of the host fractions, AMAISE's speed was better than that. The pipeline with AMAISE's speed was better than the pipeline without. So, so our contributions, just to summarize again, we developed a novel machine learning-based host depletion method that can perform host depletion accurately and cheaply. And we rigorously evaluated our approach and found that it had higher accuracy and lower storage requirements than other computational depletion methods, as well as improved the accuracy and cost effectiveness of downstream metagenomic classification methods. Thank you.
Thanks, Meera. So the next speaker that we have is Winston Zhang. He is in Nigerian lab and is currently conducting research on machine learning with applications in dentistry. Previously, he also worked at a multidisciplinary design program and works with a student team on speakers and sound recognition in vehicles. And just to remind you all, the questions will be taken at the end when all the speakers are done.
Yeah, I think that works. Hi, do I use the mic or? This one? Oh, okay, this one. Okay. Hi, my name is Winston. So I'm gonna be talking about, it's not like a study or anything, but it's just some of the research that I've done for my prelims, and it kind of like the application it has to personalized medicine. So atrial fibrillation is just like a common irregular heart rhythm condition where like irregular electrical impulses originating from the heart atrium causes the heart to beat irregularly. Usually the heart beats according to the sinus node, but in atrial fibrillation, there's different causes that can cause the electrical impulses to become abnormal. And you can see an example where the typical heartbeat has like regular spacing between each beat in the electrocardiogram. And this condition is quite common, and it can often lead to more serious conditions like heart failure and stroke. So most commonly, people use the electrocardiogram to detect atrial fibrillation. And the electrocardiogram is just it uses surface sensors on a person's body and measures electrical signals, electrical potential on the surface. And using this electrical potential, it can detect signs of, or like the internal state of the cardiac electrical activity. And this is pretty commonly known where the electrical potential as it travels from the SA node down the heart into the ventricles, it shows up as different electrical signs on the ECG surface. The atrial fibrillation event, it can be reflected on the ECG quite clearly because each heartbeat is kind of irregular in timing, and it can show up very clearly on the surface electrical potential. So generally in literature, there's two common approaches for looking at kind of the ECG waveform and kind of figuring out if a person has AFib or not. Generally, people look at two waveform details. One is the premature atrial contraction, or PAC. People call it contraction or complex, but it's basically an early heartbeat that follows directly after a normal heartbeat, and it's kind of self-explanatory, premature, right? And then this, after people collected a lot of ECG data and from AF patients, they noticed that in the ECG directly preceding an AF event or during an AF event, there's a lot of these premature contractions in the heart. And generally this can signal that the heart's about to go into a state of atrial fibrillation. And the second feature that a lot of papers use is called heart rate variability. And this, again, is looking at the intervals between R peaks in the electrocardiogram, where R peaks represent each heartbeat. And irregularity within the intervals between R peaks can signal the start of an atrial fibrillation event. So we can see that these methods have like very good atrial fibrillation performance on like publicly available data sets. And recent papers have reached very high performance on some of these test data sets where using powerful models like CNN or HRV features, they can achieve really high performance metrics in sensitivity and specificity. However, there is kind of a lack in literature of connecting these types of electrocardiogram features like the premature contractions and the HRV to kind of the underlying physiology, right? So in atrial fibrillation, there's kind of different causes that could cause atrial fibrillation. And this is quite clear to physicians who are treating atrial fibrillation in the surgery room. So if we can somehow connect these surface ECG features to kind of the underlying causes of the condition, that can teach us more about the conditions and provide a good screening tool for physicians. So here's an example of kind of the two major causes for atrial fibrillation in the heart. You can see there's ectopic foci, which is abnormal nodes driving the irregular heartbeats. So usually on the right, you can see a kind of like a 3D model of the heart surface. And the colors you can see are kind of representing the course of the electrical impulse as it goes through the heart. So when the heart beats, the electrical impulse usually starts from the top SA node, and it passes through the heart, down through the ventricles, in certain paths, right, through the heart tissue. But in ectopic foci, there's irregular nodes that kind of take over the SA node, and they start messing up the rhythm and from like an abnormal location, so that the heart doesn't beat in the correct way. So the ventricles might get the electrical impulse first, and so on. So it can cause irregularity, which causes AF. And in diagram A, so in this diagram, the top part, you can see that the blue impulse travels out from the white dot, which is the identified foci, which is causing the irregularity. And then the second potential electrophysiological cause is hypothesized to be re-entry, which can be explained as kind of like a cycle of cardiac tissue that keeps depolarizing itself. So the depolarization or the electrical impulse from the SA node it might travel around a certain node on the heart tissue, on the heart surface. And this node might be caused by scarring, or it could be caused by ischemia, where the heart tissue is like part of the heart tissue might have died. And basically, if part of the heart tissue kind of cannot transmit the electrical impulse, sometimes electrical impulses will cycle around that area. It could cycle around only a couple seconds or almost hundreds of cycles around that place. So if the electrical impulse doesn't travel in the correct motion down from the atrium to the ventricles, it can also cause the irregularities in atrium beating. And so these sources are pretty easily seen through electrograms, which is basically the physicians will put in a catheter inside the heart and map the surface of the cardiac tissue. And however, on the surface, it's not as well known. On the surface, people usually just use machine learning models to identify irregular heartbeats to kind of predict atrial fibrillation. But as to whether the atrial fibrillation is caused by ectopic foci or reentry, that is still hard to tell using ECG. And then you can see that from these two sources, there's generally physicians use what's called ablation therapy to kind of target the node that's causing the issue. So usually in both ectopic foci and reentry, there's usually a patch of cardiac tissue that might be the focus point for the depolarization to circle around, or the depolarization that's coming from that area. So using a catheter, we can intentionally induce scarring on those areas to break up the abnormal electrical conductivity. And generally in atrial fibrillation patients, after mapping the cardiac tissue and finding these targets, this type of ablation therapy has proven to be the most effective so far compared to drug treatments. And generally, you can see after a couple of spots of ablation, usually the irregularity can be transitioned back into a normal sinus rhythm. And so the current methods, they usually use invasive electrograms, which involve moving a catheter through the vein into the heart, or expensive vests for locating atrial fibrillation driver positions in the heart. But however, ideally, most patients will be collecting 12-lead ECG when they're in the hospital. And so any type of patient who has like asymptomatic atrial fibrillation, or potentially they might have atrial fibrillation in the future, they generally won't be undergoing these invasive procedures and will only be able to be screening in the hospital using 12-lead ECG. And so there's been studies that have been done where ECG might be able to roughly classify the location on the heart where the driver of the atrial fibrillation might be, and so on. And so a potential study design that I've been thinking about is patients undergoing cardiac procedures usually have these electrograms, like the basket catheter you can see at the bottom there, collected during their surgeries, but then they also have 12-lead ECG. So any type of correlation between the frequently used ECG features and then the major driver mechanisms, such as the reentry or the ectopic foci, might lead to more discoveries regarding the potential uses of 12-lead ECG for identifying patients and the underlying physiology for atrial fibrillation. And also potentially the ECG might be able to identify locations within the heart tissue where the drivers might be located. And so usually, early detection of atrial fibrillation is pretty important. So usually in normal patients, the cardiac tissue can look like that on the left. But then after you leave atrial fibrillation going for a long time, the cardiac tissue starts to get fibrosis and more spots, more and more nodes on the heart tissue become potential triggers of atrial fibrillation. And so any type of ECG prediction, such as some results from our lab previously, that can predict atrial fibrillation, can catch asymptomatic atrial patients and during ECG screening. So identifying kind of the causes of atrial fibrillation within the 12-lead ECG before the actual atrial fibrillation occurs can be very useful for precision medicine. So detection of atrial fibrillation using ECG can help physicians tailor treatment for the patient, which is kind of the point of precision medicine. And then early prediction of atrial fibrillation can give more lead time for the physicians. Thank you.
Thank you so much, Wang. It was really interesting talk on ECG, kind of highlighting the fact that how diverse this entire field can be. The next third speaker that we have is Chengpo Tang. He is a member of the Machine Learning for Data-Driven Decisions research group led by Gina Wins under the Machine AI Lab. His research focuses on developing and applying machine learning methods to solve important problems related to healthcare. His work aims to identify interesting technical challenges and propose novel solutions that are broadly relevant to AI and ML.
All right, thank you. Let me see if I can hide this. All right, good. Hi, everyone. My name is Shengpu, and I'm a PhD candidate in the Division of Computer Science and Engineering. And today I would like to introduce our work on early identification of patients admitted to hospital for COVID-19 at risk for clinical deterioration. And this is based on our recent publication in the British Medical Journal just this past February. But before I tell you all the details, I want to start by telling you a story. So in early March of 2020, the first few cases of COVID-19 cases were recognized in the state of Michigan. By late March, as our testing capacity improved, the number of cases quickly rose. Though Michigan medicine started with just a handful of cases, without intervention, it expected the number of admitted patients with COVID-19 to quickly increase exponentially in the coming days. A surge was expected in April. On March 24th, the governor of Michigan issued the stay-at-home order, and the university transitioned into being fully remote on March 26th. So this exponential increase, combined with the lack of tools and potential shortage of resources, led to tough questions. When hospitals are at capacity and run out of beds, they need to decide which patients to admit and which patients to send home. At Michigan, hospital leadership began to explore converting an indoor track into a field hospital to meet the increasing demand. Around that time, Michigan Medicine partnered with us to look at computational tools for clinical decision support for COVID-19. And specifically, tools that can help us identify which patients are at risk of clinical deterioration. If we can identify which patients are likely to deteriorate or die in advance, then we can anticipate their needs and allocate the limited resources accordingly. This may mean de-escalating low-risk patients to the field hospital or self-care, and as well as allocating the ICU beds to the most high-risk patients to stabilize their conditions. Such models could help hospitals manage a surge in COVID-19 patients. Given that, along with the rest of the world, we knew little about the disease, we took a data-centric approach and leveraged the contents of the electronic health record. Using these data, our goal is to learn to map each patient to a risk of deterioration. We can then use this risk to sort patients from low risk to high risk and then allocate the resources accordingly. Our model would also reassess the patient's risk repeatedly over the course of their admission as new information becomes available and as patients are admitted and discharged. To obtain this model, we first want to define what constitutes clinical deterioration. We wanted to capture patients that would need respiratory support, cardiovascular support, or patients that will likely die in the hospital because of the disease. In consultation with our clinical collaborators, we arrived at the following composite outcome definition, which consisted of in-hospital mortality, invasive mechanical ventilation, heated high-flow nasal cannula, and as well as intravenous vasopressors. And these constitute the different outcomes that we define as clinical deterioration. Because there were only a handful of COVID-19 cases at Michigan Medicine whose data are available, it was really challenging to train a machine learning model. Therefore, instead, we trained our model on a related cohort, which are patients with respiratory distress that are admitted to Michigan Medicine for the past 5 years. After training the model, we used all the available COVID-19 patient data for validation, which measures how well the model would perform on unseen patients. And this reflects how well we expect our model to perform in the future. Our final model is an ensemble of 500 linear models based on 9 clinical variables. But this is not what we started with. At the beginning, we leveraged the entire contents of the EHR, which consisted of over 15,000 variables. And using these many features can certainly help us learn accurate models, but it also poses various challenges in terms of debugging and integration into the EHR system, and it may lead to models that are less robust. To reduce the feature set, we adopted a hybrid approach where in the first stage, we relied mostly on clinical knowledge, clinical domain expertise to remove variables that are that do not match with their intuition. And in the second stage, we performed an iterative data-driven variable selection process to further narrow down the feature set. So to evaluate our model, we first looked at its ability in triaging the high-risk patients, which are those that will experience the deterioration outcome early in the admission. And in our validation cohort, which are all COVID-19 patients from the year 2020, there were about 20% of the patients who met this high-risk outcome. The MCURES model achieved good discriminative performance with an AUROC score of 0.8, and it significantly outperformed another model called the EPIC Deterioration Index, which is a model that was already running in the EHR system at Michigan Medicine. We also consider a secondary use case, which is the triaging the low-risk patients who are unlikely to deteriorate during the remainder of their stay after being in the hospital for 48 hours. And on this task, the MCURES model was able to flag the 5% of the lowest risk patients with a negative predictive value of 95%. So without making too many errors. And for each of those flagged patients, we have the potential to save 9.2 bed days if those patients were discharged early instead of being having them stay in the hospital. So at this point, we have a model that performs reasonably well. The ultimate goal was to package the model up and share it with other institutions. But before we do that, we want to make sure our model generalizes and it performs well on other patient populations as well. Therefore, we collaborated with the following 12 external institutions to validate our model on their patient population. In order to do that, traditionally, we would typically ask each of the institution to send us their data so that we can run our model on their data. But given the time constraint of the pandemic, this was not feasible because of the extensive data sharing agreements that's required to move these sensitive patient data. So instead of sharing data, we shared code with the collaborating institutions. We had sent our model code and pre-processing code to each of the collaborators at those institutions and have them run our model on their data using their infrastructures. And we were able to achieve this in just a couple of months. So let's look at the overall results. So the MCURES model in the end was evaluated on over 9,000 COVID-19 patients from these 13 institutions who are admitted during a 1-year period. In terms of the discriminative performance, our model performs consistently well across both the internal and external cohorts, achieving AUROC scores of around 0.8. And when evaluated on demographic subgroups of biological sex, age, and race and ethnicity, our model similarly performs consistently well across each of the demographic subgroups. And finally, across different time periods throughout the pandemic, where we look at 3-month periods in this entire 12-month evaluation period, we see that although there were some slight dips in the middle portion where there were slightly smaller sample sizes for certain institutions, in the end, our model performance actually stabilized and was also performing consistently with the overall performance. So at the same time of the external validation, we were also working hard to integrate our model into the EHR system at Michigan Medicine. And right now, the MCURES model has been running at Michigan Medicine since last year, and providers can actually now log into their patient's chart and pull up these the MCURES scores for their patients. And starting from January this year, the MCURES scores have been used by the rapid response team to drive some of their workflows. And specifically, every morning they will print out a list of the highest risk patients, and the rapid response team will proactively attend to these patients throughout the course of the day when they have some downtime. I want to conclude by highlighting a few of the main takeaways that were key to our success. First, given that we have limited data for the COVID-19 patients, we leveraged data for a related cohort that's similar enough, but also gave us more data to train the machine learning model with. And second, we relied on clinical expertise to make sure our model is robust and matched with their intuition. And lastly, we shared code instead of sharing data allowed us to conduct this extensive large-scale external validation, which helped us verify the generalizability of our model. And I want to once again mention the publication, which contain more details because I only have 10 minutes to talk through. And our code is also, code for the model and pre-processing is also publicly available online. And I want to also highlight the lead student authors, including Fahad Kamran, myself, Erkan Nautilus, and Benjamin Lee, and the 2 lead senior authors, Dr. Michael Schoding and my advisor, Janet Wings, and many more of the collaborators who contributed in various aspects to this project and all the funding sources. Thank you.
Thank you, Chengpo, especially the interesting part of data code sharing instead of data sharing. That was interesting. So the 4th speaker is Cheng Jiang. He's a member of the Machine Learning in Neurosurgery Laboratory led by Todd Holland. His research focuses on computer vision, machine learning, and biomedical data science. He's especially interested in representing learning and integrating observations from multiple modalities such as histopathological images and etc.
Oh, can you hear me? Good. Okay. So today I'm going to talk about the rapid automated analysis of skull-based tumors, their surgical specimens, and image using optical imaging and how to analyze them using that. And the talk is based on and it's based on a paper that we recently published in the Neurosurgery Journal that was actually featured on the cover this June. So let's dive right into the background. I don't think the clicker is working. So here. So here's the problem that we're trying to tackle, right? So the problem here is that we have a lot of patients with brain tumors, but we don't really know what type of tumor it is just based on the MRI image that we have, right? So the problem is that the treatment of these tumors is actually based on the diagnosis. So in order to have the right treatment, we have to know the diagnosis. And typically to get the diagnosis, you have to access the surgical specimens. So typically what happens is these patients will get into surgery and a slice of their tumor will be taken out surgically, and the specimen will then undergo a pathology workflow. But the current workflow is very slow, it's labor intensive, and it requires a lot of tissue processing, and this is not optimal. All right, so this is the process I'm talking about. Usually when we get a surgical specimen, it will get sent to a pathology lab, and the technician will have to process the tissue, right? It will have to section the tissue, freeze the tissue, and then stain the tissue, and then take a picture and send it to a pathologist. And the pathologist who is on call will have to look at the tissue and then call the OR and say, this is a diagnosis. And this whole process can take from half an hour to up to 2 hours. And during this time, the surgeon is just waiting. So can we do better, right? We want something that's faster, more efficient, and more accurate, right? So here is the alternative workflow that we are using here at the University of Michigan. So if a lot of these surgical specimens can actually image using a new microscope called stimulated Raman histology, right? And this technique will take only up to a minute to image the specimen. And this specimen, we're trying to develop the method to basically analyze these specimens using artificial intelligence and computer vision. And the goal is to get the diagnosis time to under 1 minute, right? So this will significantly reduce the surgery time and make and allow pathologists and surgeons to provide better care for the patients. And here is a panel of some of the images that we can get from a stimulated Raman histology. We can see that it highlights a lot of cellular features and the supracellular structures, such as whorls in the right middle panel in the circle, right? So, and we know that a lot of these structures correlates to the type of tumor. So let's talk about the methods that we're going to use to analyze these images, right? So many of us are familiar with convolutional neural networks, right? So in summary, the image gets passed through many convolutional layers, which we call the backbone, right? So the backbone is responsible for learning most of the visual features from the image, and it does the most of the heavy lifting of the learning, right? And from here, we hope to learn these rich expressive features for these images, right? And these features are then learned using a fully connected dense classifier. And the entire model is learned using a classification loss, right? So this loss supervises the learning of both networks. There is a better way to learn these representations called contrastive learning. And it's based on a pretty simple idea, is that these similar data should really have similar representations, right? So let's take a look at what it means in the world of computer vision, right? So when we're using this contrastive learning without supervision, right? So that is, we're learning without needing the label for each image. We can use self-supervised contrastive learning, where basically an image can go through different augmentations or different transformations. And we know that since they come from the same image, they must be similar, right? So where the loss function will try to regulate that these representations are close to each other. We can also use the labels that we have, right? So we can use supervised contrastive learning to basically figure out that different pictures of dog are similar to each other because they have the same class, right? And we can use the loss function to make sure that to attract these features and make them repel images from different classes. So with a contrastive learning framework, we will need 2 steps to train our model. We will first train the backbone using the contrastive loss, and then we will train the classifier with a frozen backbone, right? So let's take a closer look, right? So when we're training the backbone, the same image will go through different transformations, and they'll pass through the same network, right? It's the same weights that's learning from the two images generated from the same image. And we know that they're similar, so we will use the loss function to enforce attraction between these 2 images and repel everything else. And the second step is, once we have our backbone trained, we can actually freeze it, as in we can just stop learning the backbone and we'll just learn the last layer in our network, which is a dense, fully connected layer. So from here, let's take a look at the results, right? So we can see that with a supervised contrastive learning approach plus a linear classifier, we can outperform all of the existing methods in pretty much all the metrics. And we can see that self-supervised methods perform slightly worse, but keep in mind that when we are learning these models, the majority of the learning are happening without any supervision, right? The backbone is learned with no labels. To look further close into what these metrics are, we can see that the models are making a lot of mistakes in metastasis, right? So in the supervised learning and in self-supervised contrastive loss, the models are making a lot of mistakes in metastasis. And that's because that's a very diverse class. These images, the appearance of these images depends on the source tumor, where the tumor is being metastasized from. So they're very diverse. And we can see that with the supervised contrastive learning, since we're trying to enforce these representations to be similar regardless of the source, we can see we perform much better in the metastasis class. Another way to evaluate the model performance is to look at t-SNE plots. So t-SNE is a feature dimensionality reduction algorithm, basically to reduce the embeddings into a lower dimension, such as 2D, so we can actually visualize these embeddings. And we can see much better discernible clusters in our supervised contrastive models, right? Again, because we're enforcing these labels, we're enforcing these representations to be close together, depends on their label, regardless of their underlying pathology, right? These histology images are actually very big. So when we are processing these images, we will divide them into small patches. So we can run inference on every patch and create a heat map of the tumor margins. So we can see that the model provides very good margins for both the dura and the pituitary gland when there's tumor infiltration. And this is very good indicator for surgeons to tell them that maybe where is the better place to operate. And this is also one of the feedback that we got from our reviewers, because margin delineation is a very important problem for neurosurgeons when they're working with meningioma cases. So usually what will happen is they will sample at different places in the brain, and then each of these samples will then be sent to pathology to basically figure out where the tumors are, right? Because they look very similar on the MRI image. So we can see that the model provides excellent margins given the different samples being sent through the model. So this is what we have done so far. What are we going next, right? So we're firm believers in open science. So we're releasing a part of our data into the public. And one of the projects that we're working on is to basically open source our data and our code. So anyone can train similar models using our data. Another future direction is molecular classification, because in neuro-oncology, a lot of these diagnoses are moving beyond just the histological diagnosis. People are moving toward a molecular diagnosis from sequencing, and certain mutations are very good indicator for a certain type of tumor. So currently under review is one of the papers that does molecular classification with the SRH images. So in conclusion, we talked about how SRH is a rapid label-free optical imaging method that provides very high resolution digital images using fresh surgical specimens, and it doesn't require any tissue processing. And the combination of SRH and AI can achieve rapid and very accurate histological and molecular diagnosis. So surgeons and neuropathologists can provide better care for the patients. Right, that's it. Thank you.
Thank you, Cheng. So we went from host to ECG to COVID and to brain images. The fifth and the last speaker is going to be Hanrui. She is a third-year PhD student in Gon's lab. She is also part of the team which developed DCNN-based algorithm and won first prize in 2017. She is broadly interested in harnessing state-of-the-art machine learning algorithms, I am so sorry, to provide better solutions for biomedical research and healthcare.
Hi everyone. My name is Hanrui Zhang from Guang Lab. I'm honored to introduce you guys our previous project about deploying Identifies digital biomarkers for Parkinson's disease. So first of all, these are highlights for this project. So this project was based on a previous competition in Dream Challenge about Parkinson's digital biomarkers. And this project was derived from our first place algorithms in this Dream Challenge. So the other highlights of this project is that we come up with a kind of new augmentation method, which will combat the noise in an in-house environment, which will introduce a lot of randomness to the movement records and in our daily lives too. And we can based on this kind of noisy signals, we can still come up with a very accurate prediction. And second of all, we use the interpretation of this machine learning model to try to explain how our machine model allocates the kind of pathological information from the movement signals. And our method would pave a way for large-scale Parkinson training Parkinson screening in the future, because Parkinson is kind of like early onset disease. So using our daily record collected from our mobile phone every day, we probably be able to screen Parkinson from before it starts at the early stage. This come up to their study design. So all of the data in this project was collected from a mobile phone app called Empower. So this app was designed by the Sage Network, and the whole data set was used as a training data set in the Dream Challenge. So using this kind of app, you can, which can freely be downloaded from Apple Store, you can start to record your movement signal, and using those signal, we can we developed our model to detect our Parkinson's disease. So this kind of walking processes consist of 3 kinds of different phases, which like the subject will be asked to take a walking task by walk 30 seconds towards a point and stay 30 seconds there, which is called resting point, and then we'll return to the original state. So during this process, the walking signal will be recorded by the 2 kinds of sensors implemented in everybody's smartphones, which is gyroscope and their accelerometer. And the signal was presented at the right panel, which is like the series of wavelength. So based on the walking signal, we use convolutional neural network to make the predictions, because walking signal here has shared some characteristics with the image, which is there also continuous data. Based on, but we have to also pinpoint that every patient, they contain 3 different kind of walking phases, and also for each patient they have multiple. They could have multiple records. So except for using convolutional network, we use the different pooling method to ensemble all of the predictions from different kinds of records and from multiple records for each patient to make the final prediction. So I want to introduce the augmentation method. We use this to combat the noisy background in the in-house collection from our smartphones, because for everybody, our smartphone can be positioned at different starting state and different position on everybody's body. So first of all, we did contour normalize, which is to normalize the original or coordinates of the smartphone. And we also use the kind of very frequent used augmentation method, which is also used in image, is to randomly scale the magnitude or the x-axis of the signals. And then we did random rotation, because the original direction of a phone on each person's body could be random. So we don't want that kind of information to intrude our final prediction. So that's the other kind of augmentation we used. So this is our results on using both our normalization and augmentation compared to without using it. So we'll show that the learning curve on the left side will show that using the augmentation method, after using the augmentation method, it will they will delay the convergence of the learning curve, and also both the learning curve will reach slower validation loss compared to not using it. So this shows that augmentation method are currently effective for this kind of noise. And on the right side, we show like using both normalization augmentation method will significantly improve the performance of our machine learning model. So this is basically our secret in winning the Dream Challenge. We also compare different kinds of signals, like because there are two kinds of sensors implanted in our smartphones. We compare accelerometer signals and also gyroscope signals, and there's no huge difference. But we also compare like using our 3D signal, like the 3 axes of the different signals, or just do the sum square of the 3 axes. And apparently using 3D signals contains more information about people's movement, so it has better performance. And also we try to compare like using a different kind of because each person has contained multiple records. We try to compare how the different kinds of methods to pull the record prediction for each person. So we found out that by pulling the maximum prediction of each person, like the most severe records, it will shows better prediction performance than using the mean. So it probably defines that the most severe walking record for the person probably shows has more information to indicate the person's Parkinson's status. So one interesting thing is that we visualized our convolutional neural network by salience map. So we expect this kind of method will not only like and our goal is not only to get the best performing method, but we want to know why what kind of signal the convolutional network located in the walking signal. So here I put the example of the patient and the health control record here. Both the record and salience map was shown here. We found 2 interesting things. One thing is we apparently spotted the kind of symptoms by naked eye, like how the pathological walking record can be looked. So we show like the perturbed steps and the walking tremors in both the walking signals in the patient. But we also noticed that one interesting thing is our machine learning model, like the most saliency was located in the resting phase or the resting record in the patient. So we kind of feel like the resting tremor might play a more important role in diagnosing Parkinson's movement. And this kind of thing also was in accordance with results, like we compared a prediction performance using 3 kinds of walking phase: the outbound, resting, and return. And we found that using the resting record only has better performance than using the walking record. So we did some interesting downstream analysis using our machine learning model. In this in this part, we kind of like made our prediction by demographics. So we have to point out that because our data set was totally crowdsourcing from a phone app, so there are apparently bias in terms of gender and age in our data set. We show that a majority or there are three-folds more male than female in this walking record, but at meantime, female contribute more record per person than male. And we also found that there are more younger people under age 35 in this data set, which contributed to most of the healthy controls. And there are older people, they're contributing mostly to Parkinson's disease patients, which will kind of probably introduce a bias to our data sets. We kind of correlated our predictions to different demographic informations as well. So UPDRS here is kind of a classification system or to evaluate the severity of Parkinson's disease. So basically a higher score or higher UPDRS score they get, the more severe the Parkinson's disease it is. So we found that there's a positive correlation between our prediction and the UPDRS of each patient which is totally expected. And we also correlate found there's also positive correlation between the disease duration and their Parkinson's disease prediction, which is also expected because we know like Parkinson's disease is a neurodegenerative disease. It's irreversible. So it's likely the longer they suffer from this disease, the more severe Parkinson's disease gets. We found some interesting results, probably not probably we need more study about this, but this is totally from this data set. We found like there's a little correlation between the education level and the Parkinson's disease, like the higher education level that gets, the more severe the Parkinson's disease gets. But this is probably totally a bias in here in this data set. And also something is also expected between the employment status and the Parkinson's disease prediction, like the preferred retired people, because they're really older. So it's more likely they get a higher prediction of the Parkinson's disease. And for younger people, there, especially for people employee or students, there's less likely they get predicted for Parkinson's disease. So we also evaluated performance versus the training size of our model. So this data on this figure shows like how the prediction performance will increase as the increase of a training size, but will reach a plateau after about 500 individuals and 5,000 records. So this kind of shows like our training data set is adequate enough to train a good performing machine learning model. So it already reached the plateau of the maximum prediction performance here. So we also evaluate the correlation between the average record for each individual and the prediction for performance. We noticed that people with 3 to 5 records shows the best training performance here. Probably because they have the largest number of data set and also like adequate and shows adequate information for each person. So this project was previously published in Patterns, and we also have a lot of downstream study. According to this one, we use not only like the not only walking record, we also use voice record, tapping record, and memory records to do to make Parkinson's disease prediction, which is in our lab's other publications, and also our code is freely to show here. And I hope you guys do feel free to check it out. And thank you, everybody. I wonder if you guys have any questions about this.
Yes, can all the speakers come up here?
Delay before the first step. So clinically, a feature among Parkinson's patients is a hesitation in being able to make the initiation of the walk. But I don't see any evidence for that in the slides.
Yeah, we kind of noticed like in many people they're kind of, because the record didn't start at the first one, so the record, but like we didn't show like a parent's signal around the starting point. Like probably like more resting tremor shows more about that.
Okay, so you only counted once they finally started. Speed of walking. Speed of walking.
Like the resting tremors or like the resting phase in the walking kind of shows more signal, because according to our model, probably there could be like a data set bias or something. I kind of like I understand like your question, but so it shows like healthy control, they also have a cutoff starting but not much because they hit the bottom and they will take a walk. Probably healthy people, they also like start like they also hard a little bit, but
Okay.
yeah, but we didn't systematically study that.
Thank you. Are there any more questions to the speakers? Yeah, I have a question. Okay. So I have a question from Winston. When you had the ECG patients, did you also cater to the role, gender, and into the model? And how did that affect the model, if it did?
Okay, yeah. So in previous results, we did ECG prediction of AFib. And then previous results, we had age and gender matched controls. Yeah, but we did not include EHR data for the prediction. We were purely looking at the waveforms. But when we separate the training and testing set, we make sure that the age and genders are equal. Yeah.
Thank you. Are there any more questions?
Sorry, I was hoping students will ask first. So this question is to Shengpu. So you assumed there was, I guess the initial COVID emphasis was on respiratory disease. And so you used a respiratory distress model. However, later it became clear that the people who are the most at risk of dying were people, it was more like an overreaction of the immune system, which may not be in common with your initial training model. That was my understanding, at least. So have you tried after the fact? I know at the beginning when you developed this model, you didn't have data. Have you tried to make a new model that's based only on COVID? And how is that different if you did?
Yeah, that's an excellent question. So I think so due to the space and time constraint, I didn't mention one important detail, which is that even though the models are trained using the previous year's data, we actually did the model validation using a small subset of the COVID-19 patients' data. And those were excluded from the final evaluation performance. So I think that's what we stick with at the moment. And there are some ongoing efforts to like further evaluate and prospectively evaluate the real-time performance of this model because it updates the score every 4 hours, and the doctors are reacting, or like the clinicians are reacting to the scores and making some changes in their decisions. But we have not trained a new model based on COVID-19 patients only. But that's certainly something we will look into. In fact, Meera was just like also like going to start looking into some of the downstream analysis of this project as well. Yeah, thank you.
Thank you. Are there any more questions?
Hey, great talks. So I have 2 questions. So first for Cheng, you mentioned like one of your papers that you're working on is looking at the Raman histology and also the molecular typing. So are there any cases where those 2 pieces of evidence have like given contradictory assignments of like what kind of tumors you're looking at?
Yeah, so these are really different types of data. SRH is more of the histological image, so that's just the image. And the molecular labels comes from sequencing or immunochemistry. So like they're like labels you can put on the slides. So like one is the labels, one is the data. So there's really not really space for it to contradict. Does that make sense?
Yeah, it makes sense. I was curious if there's ever contrasting. And the other question was for Meera, actually. So could you like talk briefly about your model architecture, like what kind of model, what kind of features you coded them as, and like what features were the most important in the end for making that decision?
Yeah, so we used, like I think a lot of other people who presented a convolutional neural network to build AMAISE. And I think I have some details about how it differs from past work. So no past work specifically was looking into developing a host depletion method, but past work has looked into multi-class metagenomic classification. But two of the weaknesses of previously proposed solutions were the assumption of a fixed input size and also not training on data from both reference genomes and sequencing technology. And in contrast to that, we assumed a variable input size. And so we used a global average pooling operator at the end of our model to ensure that we could classify sequences of all lengths. And also we trained on sequences from a combination of reference genomes and nanopore data. And we found that, so in contrast to solutions that pad to the max length, we classified sequences more quickly. Compared to solutions that truncated to the minimum length, we improved in accuracy. And then we also improved upon approaches that solely trained on just reference genomes and only data from specific sequencing technologies.
Yeah, I was just curious if like you found like specific like motifs or just like if in the
Oh.
end like the host and
Yeah.
microbe like
Sure.
DNA sequences like had anything
Pull up
different about them.
the paper to show specifically. So one of the specific motifs that we found was, so we used a, like, I guess a similar saliency map method to determine the motifs that were most contributed to a host classification label as well as a microbial classification label. So in blue is what contributed to a host, and then in red is what contributed to a microbe. I think one of the biggest things that we found was that the 15 MERS that contributed most to a microbial classification label contained at least one CG. And I think there's like CG suppression that occurs in vertebrates. And so that was something that we found that matched with biological intuition. Interestingly enough, though, our model was much more robust to changes in percentage of CG in the sequences than existing approaches. AMAISE in purple was much more stable to changes in the percentage of CG in sequences than other methods who are much more sensitive to, I guess, the CG that they expected to exist in the host and microbial sequences.
Yeah, that makes sense. I was just curious about that specifically. Thanks.
I actually have another question about AMAISE. So y'all use it to do host depletion for, I guess, swabs. So removing human DNA to just leave you with microbial DNA. But I'm curious, would this approach also work for other host depletion problems? Like, for instance, I know someone I work with was just recently talking about trying to do host depletion for mouse xenografts, where you have human tissue on mice. So you only want to look at the human tissue sequencing. When you take a sample, you have to remove the host mouse DNA. Could you do something like that? Or would those sequences be maybe a little bit too similar since they're both from mammals? Or
yeah, so I guess we were focusing on vertebrates that are typically host versus bacteria. So we included, I think we trained on mouse and pig and human reference genomes as well as or human nanopore sequences as our host fraction. So I think it would classify mouse and host the same way. But I think that our training setup and our architecture could work well if you wanted to distinguish human and mouse. You would just have to train it to do that.
Thank you. Are there more questions to the speakers?
So this is also for Meera. You mentioned training on sequences of different lengths. So I was wondering if your sample only had short read sequencing data available, is there an effect on the performance metrics at all?
So we train on long read sequences, but you're saying if we train on short
Yeah,
read?
if they weren't if your sample only had short read sequencing available.
So I think for if it was single end short read sequencing, it wouldn't be affected. For paired end, since we only deal with single end, I guess you would have to pick one or concatenate. I think we found that performance tended to increase as the sequence length increased, but we were still able to do well on sequences of all lengths. And I think that was present in our data.
Thank you so much. Are there any more questions for the speakers? Right. Given that there are no questions for the speakers, please give them a big hand for doing this. Thank you so much.